One more.
Professor Susanna, if you want, you can rename your name. Rename.
It's okay for me, no problem.
Okay, thank you.
Yes.
You are able to share your screen? You tried? Yes, it's okay. It's okay.
Can you see? It's good?
Yes, it's good. It's good.
Okay, and you can also hear me. Okay, perfect.
Good morning, ma'am. Hello, ma'am. Can you hear me, ma'am?
Yeah. Hi. I can start when you like, okay? You just let me know.
Yes, Professor Susanna, we will, Harish sir is, Harishan sir, you are there? Yeah, we will just wait for 5 to 10 minutes and then.
Okay, okay. Let me just know, okay?
Then after that, we will start. We will just wait for the students. Yeah, thank you.
Good morning, ma'am. Shall we start the session? We have around 20-25.
Yeah, Harish sir just
25.
said we will wait for 5 to
Ma'am,
10 minutes and
Can you
then,
hear me, ma'am?
Harishan sir, just informed ma'am that we can wait for 5 to 10 minutes and then start. Should we?
Yeah, for me it's okay. I start when you would like, okay?
Ma'am, uh, I think we can start the session.
Okay,
Okay, ma'am.
can you hear me? It's okay?
Today also we are going on YouTube live for GIAN purpose. We are also live streaming. Uh, I think we can.
Hello?
Yeah, Dr. Harikrishnan is saying we are at the same time we are doing live streaming in YouTube.
Okay.
Okay. Yeah.
It's okay? Can I start?
Yes. Yes. please.
Okay. So good morning to everyone, and thank you to and welcome to this second day about our course about bio-based and bio-inspired materials. So today we will continue with the bio-based materials. So the second polymer or biopolymer that we will discuss is starch. So starch and its derivatives, and I think that so I will do the first part, and then Professor Sabu will talk about PLA, so one of the derivatives of starch. So here you have the outline for today. So first I will discuss with you about general aspects, then the physical and chemical modifications of starch. And in the last part of the course, I will talk about nanostarch. And finally, I will finish with applications of starch and nanostarch. And then you will learn also about PLA. So again, the learning outcomes come as I did yesterday. So for me, again, what is important for you is to be able to discuss about biopolymers, the importance of biopolymers, to know what is the role of this biopolymer in nature, and then you can apply it. So you can extract them and you can apply it in material science to do the materials of the future. And for sure, to do it well, it's necessary to know a little bit about the each biopolymer and its properties. Okay. So what is starch? Starch is a polysaccharide. So yesterday we discussed a little bit about polysaccharides, and so it's a polymeric carbohydrate composed of glucose. Later in the lecture, we will see how these molecules of glucose will be assembled together to do starch, that it's different from cellulose for sure. And these molecules, they are linked by glycosidic bonds. So where can we find starch in nature? So we can find it in green plants and also in agriculture raw materials. And what is the role of starch in nature? So we see it yesterday. So actually, starch is the molecule that store the energy in plants. And like I did for cellulose, so I also propose to you to see what is the importance of starch for us, for the humanity. So for sure, starch is the one of the main caloric resource for human. And the main sources of starch are cereals and vegetables. And as you can see here, wheat is the first source of starch and the grain the most consumed by humans. And after to extract starch from the plants, so what is very interesting is it's a white powder and doesn't have, or is a tasteless and doesn't have also odor, and it's insoluble in coal, water, and alcohol. and how we can degrade, or the animals can degrade starch. Actually, it's with an enzyme that calls amylase, that it's found in our saliva. Here, just a little bit of story about starch. So the first use of starch, it was in the elaboration. So yesterday also we talked about it, about the papyrus, as a support of a way of communication. So here, starch, it was also used in papyrus to do this kind of paper. And it was also used like powder hair, so to increase the volume of the air. And it's still used with this kind of application. And in China, for instance, it was used to coat some documents, to protect the documents. And they were able to do starch in the form of powder. And in the Middle Age, so we start to use starch in laundry to give some stiffening to the fabrics. And later, so and still today, it's used as color printing. It's used in textiles, paper, still being used in paper industry. And also it was discovery the way how to convert starch into dextrin. So it's we can do it by eating starch or by acid hydrolysis. And more recently, we start to talk about nanostarch and to extract nanostarch or do nanoparticles of starch. Here it's in Europe, sorry, but it's just some numbers about the production of starch in Europe. And it's just to show you that maize and wheat are the most sources of starch in Europe. And so what are the sources or origins of starch? We can have it from cereals like corn, wheat, maize, rice, legumes, so and also fruits, banana, roots, etc. So these are the some of the sources of starch. So starch is how it works. So starch is found also in the photosynthetic organs of plants. So during the photosynthesis, the plant used light energy to produce glucose from the carbon dioxide. And this glucose is used to generate chemical energy, or to, as I told you yesterday, so is a compound that it's the starting point to do other kind of compounds like lipids, proteins, and cellulose. And starch, it can also be stored in the form of starch granules, so that we can call starch granules or amyloplasts. And we have here an image of an amyloplast from potato cells. Okay, you can see here in the optical microscope. So yeah, we can call both names, starch granule or amyloplasts. So these starch granules can have different size, and it's like the fibers yesterday from cellulose. It can have different forms and size, and depends for sure the source. So you can see here, for instance, the starch granules from potato, they are very big regarding, for instance, corn or wheat. And also the form, as you can see in the different image, they are completely different. So we can have different forms and size of the starch granules. Now I have two examples of grain, so wheat grain, and to show you where starch is stored. So it's in the endosperm, so it's this winter part of the grain. And here, again, we can see a microscope image of the wheat starch granules. and you can see they are circular, but they are like spread. And if you see the corn grain, so again, starch is stored here in the endosperm, so in the inter, and this part in the middle of the grain. But here you can see it's more like so they are also spheric, but they are more in 3D dimension, so they are bigger than the other. Okay. And like cellulose, yesterday I show you the multi-scale the multi-scale structure of the cellulose. So from the cellulose chain to the tree. So today I show you, so starch, we will see next that we have two polymers that give starch, but here it's the same thing. So it's amylopectin and amylose, and these molecules, these polymers or biopolymers, they will be assembled together to do this bouclets of starch. And we have a mix of amorphous regions also, like with the cellulose, and crystal regions. And then these bouclets, they will be organized in order to compose the grow rings of the starch granule, and with, again, the zones that are amorphous grow ring and crystalline lamellas, and this will form for sure the starch granule. Okay, it's the same thing. We have also this multi-scale structure. So like we did yesterday for cellulose, so what is starch and what is the interest of starch? So starch, as I told you just before, it's a mixture of two homopolymers, so amylose and amylopectin, and so they can be so amylose, it's more linear, and amylopectin, it's more branched. So, and like yesterday when we learned about carbohydrates, so, or the monosaccharides, so starch, we have more, it's alpha linkage 1,4 or 1,6. We will see it in the next slide. And this, it will do that the starch has this helix with 6 glucose units per turn, 1, 2, 3, 4, 5, 6. And it depends how it's done. So we can have the amylose or the amylopectin. And here we have, regarding the structure of these two polymers, so the differences between these two polymers. So amylose, as I told you, the configuration of the chain, they are more linear, and amylopectin, it's linear and branched. The type of bond, so this is also important. In the amylose, for instance, we have only alpha 1, 4. So as you can see here, so 1, 2, 3, 4. So we have 1, 4 glycosylic bonds. And in the case of the amylopectin, so we have here, so most of the linkage, they are 1, 4 again, so 1, 2, 3, 4, but we also have around 5% that is alpha 1, 6. So you can see here, so here you will have carbon 6 that it's linked to a carbon 1 of the other chain, and this will do the branch of this polymer. Sorry. So regarding for sure the number of branches in amylose, it's very few, and in the amylopectin it's important. And we can also see that the molecular mass, it's in Dalton, it's in the case of amylose, we have it's smaller than for amylopectin. And in terms of the glucose units, You can also see that we have, as we can see by the molecular mass, that it's different. It's smaller for amylose. So still in the amylose versus amylopectin, the ratio of these two polymers, like yesterday the cellulose, it depends of the sources, sorry, of this starch. So for instance, in rice, we have only about 17% of amylose and 82% of amylopeptine. But, and for instance, in the waxy corn, we don't have amylose. We have only aminopeptine. So you see, yesterday it's also someone asked me, how can we have, okay, well, from which source we can have nanocrystals of cellulose or this kind of thing. So it's the same with the starch. So it depends of the source. So it depends. We know that with the amylose, we can have more crystalline zones. So in this case, if you want nano crystals from starch, so you need to see what is the origin or the source that has more amylose. Okay. So, and for sure, this configuration and this kind of polymers, like for the synthetic polymers, so it will do that we have different properties. In the case of amylose, so it's more crystalline in nature, like I just tell you. And this, it will do that the sources that has more amylose, they are insoluble in cold water and more resistance to the digestion, because it's the same thing. So with enzymes or with chemical reactives, so we need to have the linkage or the functional groups that are available for the enzymes or for the chemical reactives to penetrate and to hydrolyze or to modify or to degrade the molecule. So for sure, if it's crystalline, it's more difficult to degrade or to do the digestion of starch. In the case of the amylopeptin, so it's soluble, it could be soluble in cold water, but also in hot water, and it's easily to digest. So this is regarding the properties. So in general, for starch, regarding the physical chemical properties, so this is the chemical properties, so are those that the properties exhibited without any change in the chemical structure of the starch. And the two main physical properties that we need to know about regarding starch is gelatination and retrogradation. So we will see now. I have this figure that I found. You have here the link, then I will give you the supports of the lectures. So you have here the link, and you can see a general view about gelatination and retrodegradation or crystallization. So we will see now more in detail what is this. So the gelatinization of starch, so like the name give us an idea. So we will do a gel, like a gel to starch. So, and to do it, the most important physics thing that happens is that the granules or the starch granules, they will swell up in the presence of water and heat. So it's the conversion of the semi-crystalline granules of starch to disperse in amorphous molecules. So this process is irreversible, and because it's breaking down some intermolecular bonds. And we can do it with water, so it works very well with the water, but we can also to use some organic solvents. Now I have here more in detail. So here it was just to show you the gelatinization, and here I have a more detailed graph with regarding the gelatinization of the starch. So here in the beginning we have starch granules that are made of amylose and amylopeptide. And we can see linear and branched chains. So when we put water, even if it's cold water, so the water, it starts to break the amylose crystalline and disrupt the dialysis. And the starch or the granules, they start to swell. And then if we put more water and if we heat the water, so the amylose starts to diffuse out from the granule. And then we have the granules that are now mostly containing now amylopectin, and they are like in because for the amylopectin has its branched, it's more difficult to be diffused from the granules, and we have like a matrix of amylose, and inside of this matrix we have the granules collapsed with amylopeptide. So now I have so once you do it, if we characterize it, so we can see the different properties of this modification of the starch. And we can start by the crystallinity. And here, so you can see the three patterns. We have first the native starch. So you can see the peaks that are related with the crystalline zones of the starch, and they are characteristics from the starch. And then with the partial gelatinized starch, you see that these patterns, the intensity, they start to decrease. And when we have the completely gelatinization of starch, here you have a pattern. So it's like you see this pattern. It means that it's amorphous polymer. And this is not for only for this kind of pattern. It's not for starch. You can also have the same kind of pattern with chitosan, with polulan, because they are amorphous biopolymers. So you can see these kind of things. And if we look at by SAM, the microscope, so the morphology, we can also see, and this is very beautiful to see how we transform these starch granules. So you see, in the beginning, we have the native starch granules, and then with the completely gelatinization of starch, you see, you have this, you don't have no more these granules, you have like a matrix of starch. So regarding the viscosity, how can we change the evolution of the viscosity during the gelatinization of starch? So here again, around the 30 degrees, you have the starch grains, and once you start to increase to 60, 80 degrees, so then, you can start to see the starch granules that they start to swell. And then you have around 90 or 100 degrees Celsius. So you have the maximum of viscosity of your solution of starch, as you can see here. And then if you start to so then you have the completely gelatinization. And then if you start cooling your, I know that some of you work a lot with starch, so you know it. If you start to cooling, so then we have what we call edging, or the recrystallization of the starch, that is the retrogradation of starch, as you can see here, okay? So it's the recrystallization of starch in which the amylose and the amylopectin chains, they start again, or they try again to be organized way and to be crystalline. So regarding the retrogradation of starch, so it could be affected. Regarding the amylose content, for sure, because this is important, because it's more easy for the amylose to be again well orientated and in the final product that we want. It's more difficult for the branch to do this recrystallization, and for sure it depends of the source. So amylose contents, and also the length of the amylopeptide change, the density. So the quantity of water or that you have in the paste. So what you are doing. Also the storage conditions that you put your final material. And for sure the presence of other compounds. If we put, for instance, some plasticizers or other kind of, if we did before some modifications to starch, etc. So we talk about the physical chemical properties of starch. Now let's see regarding the chemical properties of starch. So I like cellulose. So like cellulose, it's glucose monomers that we have. So we can see here where we have R. It means that there are the three OH groups that we have that can be, we can functionalize, we can do some modifications to starch. And so we can also do some cleavage to the glycosidic bonds here between the different links of the chains. And now we will go through the other part of the second part of our lecture, when I will talk you about the physical and chemical modification of starch. So why to modify starch? The same like for the other biopolymers. It's because we want to improve the functionality of this polymer. We want to give better dispersibility of the polymer in the matrix, or we want to increase the compatibility of this polymer with different solvents or other matrices, etc. So the aim is always the same. So here I found it in a publication, and we have here the main methods of starch modification, and I think that it's a very good summary about the starch modifications. And as you can see, it is divided in two parts, so physical modification and chemical modification. And regarding the physical modification, we can have thermal modification. So we will talk a little bit about it, and no thermal modification. So as mechanical treatments, I also will show you an example. pH treatment or pressure treatments, ultrasound, sonication, etc. And so I have some examples later in the presentation for you about the physical modification and about the chemical modification. And I think that, yes, we will start by chemical modification. So oxidation, hydrolysis, esterification, cross-linked So you see, it's almost the same kind of modification that we can do with cellulose. Yeah, so for instance, I have an example about the cross-linking. Actually, here it's a mechanism of covalent. It was to do a covalent interaction between starch molecules. And to do it, we used a derivative of sodium, of a phosphate, or we used sodium 3-metaphosphate, or other kinds of phosphate you can use. And actually here it was to replace the hydrogen bonds between the starch chains. And in this way, we have covalent chains, and we can to increase or to improve the resistance of starch. And this kind of modification, it's used for starch derivatives that are used in food applications. Another kind of modification. So I told you about the dextrine. So we can have, we can produce dextrins from starch. So just by eating around, I think it's if you eat around 160 degrees Celsius, I think it's the temperature, the good temperature, or if you do some acid hydrolysis, so you can have dextrins. And if you modify these dextrins with enzymes, you can have, for instance, the cyclodextrins. And the cyclodextrins, they are really very important for food and pharmaceutical applications, because you can see here in the middle, actually, we can put a drug or a nutrient or a food substituent, and we can, it's used as a drug delivery system. And it's really used a lot in ophthalmology for the drops eyes with the drugs inside, because you see, has this size, and then we can have from let me see, 1, 2, 3, 6, 1, 2, 3, 4, 5, 6. I think it's between 4 and 7 glucose that we can have to do different size of cyclodextrins. And they really are very interesting, for instance, because the solubility, you know, it's more difficult, and so you can retain more time the drug in your eye. And for sure with also with the enzymatic way, we can do some oligosaccharides and modify the size of by modifying the size of our starch. So regarding the physical modification, we have so this kind of eating and mechanical sharing, and it's an extrusion machine. I don't have the details of how they do it, but actually it's to generate starch ingredients with that can be instantaneously soluble in cold water. So actually we break the links between the different chains of the starch. We do it very small, and then we can use it in cold water. What is interesting to do it in as a gelling and a thickening ability for foods. So I have here some, you see the instantaneous soups, they use it, or to do some bakery mixtures, etc. So it's used as a thickener in food, and also to do some pasta. So this is one way that we can modify physically starch. And the plasticizer starch, so that I think that you know, we can eat the starch until 100 degrees Celsius, and then we can do different amounts of water, and we can use different plasticizers. Then we cool it, and then we can have a material that can be flexible or not. So to give you some ideas of works that we did here in the lab with starch by plasticizing starch. So we did different kind of composites. So in one kind of composites, we used vegetable cellulose. So as you can see here, is from a hardwood that we used, and to do some materials, and what others by using material cellulose. And here you see the starch granules. And so we did it by using 45% of starch, 25% of water, and some glycerol to have some flexibility then. And cellulose, we changed the quantity of cellulose. So here I have an example of using 5% of cellulose. And we did it by using this melting equipment. And you can see here, so then we had some composites using bacterial cellulose and vegetable cellulose. And you can see here, so because one of the disadvantages of starch is that the mechanical properties, they are very low. So to increase the mechanical properties of starch, we use these two sources of cellulose. And as you can see, and as I explained you yesterday to you, as we have very small nanofibers with this bacterial cellulose, and they are very crystalline, very well organized. Actually, we were able to increase the Young modulus of the materials that were done with bacterial cellulose. We also did some materials. So two kind of materials to do a thermoplastic starch. So in the left side, we analyzed, so we did by solvent casting. So yesterday I also show you different ways to do materials. So I discussed it with you about solvent casting. So we first dissolved the starch in water at 90 degrees. And then we incorporate different forms of chitin, so nanocrystals in one kind of materials and nanofibers in the other. And then we just let dissipate or the evaporation of the solvent, that was water. And in the other method, so we use this melting way to do. And we did the same thing. We used nanocrystals and nanofibers of chitin to see the differences. So, and here, actually, we had two objectives with this work. It was to increase the to increase the mechanical properties that I will show you in the next slide. But it was also to give some antimicrobial properties to starch, because starch, like cellulose, so the glucose doesn't have, so we don't have amino groups, for instance, with antimicrobial properties. So yesterday with cellulose, it was the incorporation of silane derivative with the NH2 groups. And here what we did, it was to incorporate chitin that has N2H groups to improve the antimicrobial properties of starch. And we can see here in the right side, So in this case, we didn't use bacteria. We used fungi to see if the fungi they grew at the surface. So, and you can see here in the starch film, with the time it was almost covered by the fungi, and with 10% of chitin nanocrystals or chitin nanofibers, we can see that the film, it's not, it's like it's protected. It's not the fungi, they can't really cover all the film. Regarding the mechanical properties, as it was expected, so we found that with chitin nanofibers, as they are longer, so they have better, they show it to have better mechanical properties regarding the young modulus. And for the method that we used with casting evaporation, but we also found the same kind of properties with the materials that were prepared by melt mixing. Okay. So I think, let me see the hour. Okay. So I will continue with the nanostarch and the applications of starch. This is the third part of the lecture. And then at the end, as we did yesterday, we can discuss, and we can you can put your questions about this part, okay? So about we will start with nanostarch. So about nanostarch, also I propose you, it's a review paper. It's from 2010, but is very good because it's really show you, as you can see here in this schema, the different ways that we have to have starch nanoparticles or nanostarch forms. For sure, there are an evolution on it. And I also, then I have more recent publications, but this is a good point as a starting point for you if you are interested in nanostarch. And then for sure, now it's 13 years more in evolution of the research. So for sure you will find more recently papers about it, and also reviews, but this, I think it's a good starting point, and it is because of this that I put here to you. So we have three main ways to have starch nanoforms or nanostarch. So we can have starch nanocrystals, as you can see here in the bottom part. So using hydrolysis, we can have nanocrystals. I will show you then more details about it. If we do some precipitation, we can have what we call starch nanoparticles. And if we do mechanical treatment, so like I told you before, for instance, to have not nanoparticles, but in order to have to be able to dissolve easily starch, we can have, so with mechanical treatment, what we call nanocolloids. Okay, so let's start by nanocrystals. Oh no, before, sorry. Why are starch nanoforms important? Is so this is another publication, and it's this is from this year. So where we have the properties. So it depends of the content of the amylose, as I told you before, the size and distribution of the particles that you will have, the starch crystallinity, the thermal properties, etc. And the kind of applications that we can do with the starch nanoforms, it's for sure using any emulsions, also to do bioplastic fillers, and also to use as the encapsulating agent. Okay, so starch nanocrystals. Here you can see these images, microscopic images, where we can see a longitudinal view of the starch nanocrystals and the planar view of the starch nanocrystals. And just to remember you regarding the multi-scale organization of starch, you see, so what we are looking here in this image is this part that is this crystalline parts of the starch. So to prepare the starch nanocrystals by acid hydrolysis, so we need to have in account different kind of factors. So the starch concentration, the acid concentration and type for sure, the time of the agitation and the temperature, and also the kinds of the agitation or the number of rotation per minute. Okay, so this is very important to take in account to do your chemical hydrolysis. So here I have some from the same from this review from this year. So I have some it's also very nice because it's about all the kind of types of that you can do starch nanoforms. So regarding the acid hydrolysis, you can see, so for regarding the conditions preparations, you can see here. So they used hydrochloride acid, and the concentrations of the acid, depending of the acid, as you can see, it's different. They also use different kind of temperatures, and you see it's from several days. So it's for several days. Here, for instance, we have between 5 and even 10 days of acid hydrolysis. So it's really very long process. And then you can have here, you can see here again, so it's two kind of morphologies of starch, rice and wheat. And then you can see also the kind of nanocrystals that you can see here in the image by TEM. Okay, the different morphologies. Nanoparticles. So how can we do nanoparticles? It's by nanoprecipitation. So you can first put your you can first to do the gelatization of the starch. So with the glycerol and water and the starch, so you have your starch dissolved in your solution, and then you can put something like drop by drop of ethanol, and by mixing it, and you can have nanoparticles of starch. And for instance, here you have, this paper, it's from last year. It's also a nice paper to see how to do it. And actually, you have nanostarch in the matrix of starch. And then what they do is that they do a film just by casting this suspension, and then they have a film. Here you have this image of IFM. They have the film where the matrix, it's the matrix and the nanoparticles are starch. Okay. So again, a little table doing a summary of some nanoprecipitation. So using ethanol, you can see it's really used as a solvent to do the precipitation. It was already done with starch, potato, tapioca, waxy corn, etc. And then we can also do something using acetone and also butanol. Okay, so these are alcohols that we can use to precipitate starch and have what we call these nanoparticles. So for the preparation of the nanocolloids, it's a mechanical method that we can do it. I have here, let me see, yeah. I have here just two examples. So usually it's the less complicated, the less expensive, and the less time consuming. And for sure, as we are in the time that we are trying to have the less and less the less and less chemical reactives, for sure mechanical way it's good, but the problem is that we have a lot of energy consuming, so we need to do the balance regarding this how to produce the nanocolloids of starch. Okay, so I have two examples here. So the ultrasonication, so you know this kind of method. So the ultrasound generates mechanical energy, and it will disrupt the linkage between the starch molecules. And so we have small particles of starch. And if we use gamma irradiation, for instance, so in this case, it's the dispolymerization again of the starch chain by breaking the glycosidic bonds. And in this case, we also can have small fragments of starch. And again, I have here this the table regarding different kind of, for instance, for the gamma irradiation. So we have different we have the dose that they use to do it. We can also see that it was already done with different kind of sources of starch, corn, cassava, oats, etc. And you can see here, the gamma irradiation, but also the high pressure homogenizing. I like this technique. I also use it to have nanoparticles of chitin and cellulose. So this technique, it's good, but you need to take attention because it's a small valve where you do the injection of your suspension, and sometimes you have some problems because it's blocking this. It blocks the valve, and yeah, ultrasonic. Okay, so starch applications. Regarding the starch applications, I have here this graph showing that for sure starch it's for food, it's a lot of kind of applications. So 23% for drinks and confectionery, 5% for bakery, 15% for other kind of food, you know, prepared food, 5%. And then we have also a big part that it's for paper. As I told you, we used it a lot as a additive in a paper, and around 7% for pharmaceutical applications and for chemicals. So let's see it more in detail. So here it's again a list of kind of the applications that we can have, and I have here some numbers. Again, it's for Europe, but 8.9 millions of tons of starch and its derivatives, they are consumed each year in Europe. So you see, it's really big use of starch. And for instance, is if you look when you go to the supermarket, and if you look all the additives that you have in food that you buy in the supermarket, you have a lot of codes. That's, for instance, the E1400, it's dextrine. You see, so you have these codes that are all related. So the E14 with starch and its derivatives. So it's the international number system for starch. So you have here, you see, we talk about phosphates, these starch phosphates. So actually, as I told you, it's an additive for food. So you see, it's also appearing here, a lot of them. And here in this table, I just summarize some chemical modifications, typical chemical modifications process and their application in food. For instance, when we do some acid treatments to do hydrolysis, for instance, it's to reduce the hot paste viscosity, as I told you before, and to improve the texture of the properties of the starch. It's because we want to use it, for instance, for some gums and jellies that we used in food. For instance, the esterification of starch by doing carboxymethyl starch. As yesterday, you remember, we talked about it with cellulose. So then we can have cold water soluble starch, and this is used to do candy and foods and sweets also. And for instance, the cross-linked, as we see, it's used to do some sauces, soups, bakeries, etc. So you see here you have this table. I think it's good if some of you are working in food applications or also cosmetic. You can find the properties, so the kind of modification and the properties and the kind of application. And now I have here, so one of the main applications of starch is the edible films. We try to do some of these kind of applications with our students. They are in polymer, introduction to polymers. And so we did we did the same thing that you can see here. So two kind of edible films. So first we prepared some films by casting or evaporation casting, and then we did some bags to put our fruit inside. And the other, it was by immersing. So we do the solution of starch, and then we put our apple inside, and then again the solvent, it was evaporated, the water. So, and we have a coat in the apple, and this it was to for instance to increase the time of conservation of the fruits. And it was working. It was really very nice. And we did the same thing, but putting some chitin nanocrystals inside. And what was amazing, it was then with the chitin inside, the nanocrystals, we observed that we were able to conserve more time. Because we were looking for the antimicrobial activity, but for sure starch doesn't have. But even only with this coating, we see some progress without in comparing with the fruits that it were not coating. And what was amazing, it was regarding the quantity of water that was diffused from the fruit. So we lost less water in the fruits during the time that were coating with this kind of so with the starch and starch nanocrystals, chitin nanocrystals. So another application of starch is this kind of packaging for food. So and it's we can do like expanding starch. I was not able to do it. I never do it. Maybe you did, and if you already did it, you can explain me, because I would like to learn about it. And so this is good because there are a lot of synthetic polymers that we are using to do this kind of materials: polystyrene, polyethylene, polyurethane, as you know. And so if we can replace these polymers by starch to doing it, so it will be fantastic that if we can do it. And yeah, I just finished the course, and I have here some of the references that I used to prepare this course. And I don't have here my publications, but you have in the presentation all the information about it. And if you are not able to have the publications, I can also to send you. So now we have time to discuss, if you like, before you learn about PLA. And we can use the same method than yesterday, if you like.
I request everyone to ask some questions, please. Please ask some questions.
If you don't have, I have. For you. Some of you work with starch? And if yes, for to do what kind of materials? We can discuss. If you, okay, I see that there are some up. Okay, can you see this is very good again. So inside, I don't know, can you see the question? Are you able also to see?
Yeah,
or
yes,
should I? Yeah,
see.
okay,
Yes.
okay. So I'm very happy with this kind of comments, because it's really something that we are moving on. And I don't again, for this kind of questions, I don't have the answer, but so I'm in discussion with some of colleagues that are working in machine learning and artificial intelligence. For instance, in June, I spent a week in MIT to learn a little bit about it. And not for me, the course, it was important regarding the part of bio-inspired and biomimicry materials that I want to do. But I think that this is the future, and for sure that machine learning will help us go in this direction. I don't work with this, so as I told you, I don't know how to answer to your question, but it's very pertinent. It's very good question. But from my side, for instance, what I can tell you why I did this course in MIT and what I'm waiting for machine learning. So as I told you yesterday, I'm trying to understand so how is the architecture of this, the DNA, proteins, lipids, these carbohydrates, how they assemble together in nature, and also with evolutionary perspective, because and I'm trying to have to characterize different kind of materials and to give this information with the properties that I want to machine learning, and then to understand first to understand with machine learning these properties and this architecture, and then to try to do better materials. But the way, so this I don't know. For the moment, I don't know. But it's something that I'm starting to try to give this direction to my research. And I'm really very happy that you think about it. Yeah. I think so. Yeah, for sure. Yeah, I think that that we can have for sure. I also can give you the name of he calls Marcus Beher. It's from United States. I don't know if you know his papers and what he's doing in with machine learning and biological materials, but it's amazing. And he used a lot of regenerative artificial intelligence and this kind of algorithm software. So I can give you some information about it. But for me, it's very, I'm just starting. And I'm trying to have a collaboration. So with him, about the part of the structure of biological materials, and here at UPPA, then for the part of to do materials and nanomaterials with this information. But because one thing, it's that it will be very complicated. It's like with this is again a technique that or a technique that I don't use. It's 3D printing. But you know, the idea to do from the nanoscale to macro scale with the 3D printing using the same techniques that nature used, I think it's something that machine learning can help us. Good question. How nanostarch work as antimicrobial agent? So I never work with nanostarch as antimicrobial agent, but we did some experience with nanocellulose, because you see, nanocellulose chemically doesn't have functional groups that has this property. However, we did some with bacteria and with fungi. We did some works that we proved that the nanosize, the nanosize form of nanocellulose. Again, not nanostarch, but maybe with the crystals, we can have with nanostarch, we can have the same properties. It's that the nano size of the cellulose, it was able to penetrate the membrane of the cell, so of the fungi or the bacteria, and have a negative influence in the bacteria or the fungi and the microorganism dead. So with nanocellulose, we have a paper on it. With nanostarch, I never work doing it, but I believe that the crystals ones, maybe we can have the same properties. Oh, so for the question, sorry, about nanostarch sensitizers and photodynamic therapeutics, I don't know. I never work on it, and sorry, I can't answer to your question. Then I can help you if you like to search in the literature or something and help you. But today I don't know. I never work with it. So how can we preserve the nanostarch particles? This is also a good question. It depends of the kind of nanoparticles that you use. So the thing is, when you do by precipitation, what happens is that they collapse with the time. Okay, so this is difficult. The best thing is that you prepare it and you use it. For the nanocolloids and the nanocrystals, for this kind of things, we just prepare our suspension, or extracting the starch nanocrystals in water, and so we used acid hydrolysis to have our starch nanocrystals, and then we do dialysis and centrifugation to have a pH, neutral pH. And then what we do is that we try to put it in the fridge as a suspension. Actually, we don't remove completely the water, because then to homogenize it in the material that we want to use, it's better. Okay, we do like this. And for the nanocolloids, it's the same thing. For the precipitation, I think that the particles, they will aggregate, because it's different kind of nanoforms. Okay. So regarding the question, can we cluster all kinds of different polymers? I think that you are talking about the biopolymers to create nano-level structures and extract its different properties. Probably. So I have experience with bacterial and antibacterial and antifungal, with parasites and viral. I don't know. I never work on it, but I don't know. I don't know. so I can't answer you to your the last part of your question. Sorry, I need to check it. I don't know. So Tony, so in the present scenario, which one is the novel biopolymer? What do you mean biopolymer in general? Yes. So again, for the biopolymers, you can do so. You can do if it's a biopolymer, again, to do the nanoparticles and the nanoforms. So if you want to do nanoparticles or nanoforms from biopolymers, if all of them are able. So regarding the nanocrystal parts, it depends if the biopolymer is amorphous or crystalline, or amorphous or semi-crystalline, because there are some polymers that are semi-crystalline, like chitin, cellulose. Okay, so in this case, you can separate by acid hydrolysis the nanocrystals and have nano objects. Okay, but there are other polymers or biopolymers that are amorphous, like chitosan, starch. So starch, it's also so you can also to have both of them. Sorry. So in this case, you need to prepare nanoparticles. So by precipitation or by using, we do a lot of chitosan nanoparticles by using ultrathorax or microfluids system. It's also work. And then for sure, you can try to see if it's antiviral, antimicrobial, or antifungal, okay? But as I told you, my experience, it's only with bacteria and the fungal. So then I lost some questions. So in the present scenario, which one is the novel biopolymer? In general, you know, we will talk about it in lecture 4. There are a lot of new biopolymers that are being discovered, marine ones, because as it's until some years ago that we are able to go deep in the ocean with and to bring some samples at the surface and analyze it. There are new marine biopolymers that we are discovering right now. I have some videos, and one of them, it show it for you. The thing is, it's in French, and I'm looking. I know that there are a translation of this video, and I'm looking, but we will see it in day 4 and day 5. Okay, about the new biopolymers. Good question. What are the challenges in starch research? Wow. I would say that is the modification without using using green chemistry or functionalization of starch. You see in the table that I put you, because starch it's used for food applications. So yeah, if you sorry, I'm lost. Oh, yeah, if you are able to do it with green solvents, or I think it will be a good way. So have you worked in magnetic cellulose nanocrystals? No, never. I never work on it. No. No. Mm. What are the applications? So the applications was so with I don't know if I understand your question. Fabrication of asymmetric nanostarch reinforced chitosan. So you mean that starch is reinforcing chitosan matrices? Um, Sorry, there are a lot of questions right now. Give me a little bit of time. So sorry, I don't understand the question. If the applications, fabrication of asymmetric nanostarch reinforced chitosan. So if you can give me some other keywords, please, and then look how. Oh, for the how I look food packaging and increasing food's shelf life properties using biopolymers. For sure, this is for me. It's for the one using one used packaging. I think it's the best thing to do. It's using biopolymers. But the thing is that we need to use biopolymers that can be solubilized in water. It will be the best thing to do it. But yeah, I think this is something very important, and that we should still continuing doing research on it. How to use biopolymers for food packaging, for sure. Which one is the best source for scientists? Oh, precipitation nanocrystal, precipitation acid hydrolysis, or colloids. It depends what you want of your what you're looking for as properties for your for instance, if you use hydrolysis acid, it means that you will have nanoparticle or nanocrystals of starch, so that can have some mechanical resistance and properties. But if you use precipitation, what means is that it's amorphous starch that you are doing the precipitation. So even if there are what we call there are some structuration of the starch chains, it's not the same like in the pristine crystal. Okay, so the properties, it will be for reinforcing, for instance, it will be not the same. So it depends what kind of application you want. But for instance, if you want to use it for drug delivery, so in this case, is better the precipitation one, or using, for instance, this is, as I told you, for chitosan. We do chitosan nanoparticles for drug delivery. In this case, we use ultra-2rex, for instance. Okay. If I write a proposal, which novel polymer I take? It depends what you want with your proposal, but if you like, we can discuss about it, okay? Because it depends on what you want to do. So it's because of this that you ask me what is the novel biopolymer in your question before. It depends. Really, it depends of. So then for the marine biopolymers and new ones, and we will see it later in the week, there are some marine polymers that has sulfur. So it could also be interesting for some applications. Yeah, for biomedical applications. Which one? Oh, okay. Okay, so for biomedical applications, it depends if it's for drug delivery. So as I told you, it's better precipitation, or to do the thorax. So and if it's for to do a scaffold for regenerative medicine. So if it's starch that you want to use to do the matrix, so it's just to do a solution of starch. Or for instance, for medicine, we used for regenerative medicine, we use a lot of chitosan as matrix, and then inside we put or collagen as also matrix, and inside we put nanofibers or nanocrystals of chitin. In this case, why we are putting nanocrystals, so I think I will discuss about it tomorrow, but I can tell you now. So the chitin is because nanocrystals is because cells, they need some rigidity in the scaffold. And so we put the nanocrystals to have some rigidity or mechanical rigidity in our scaffold, but also because chitin has antimicrobial properties, and it's good for medical applications, as you know. So it depends if these biomedical applications that you are putting here is for drug delivery or for skin or for medicine regenerative, okay? Oh. I don't know. So Kumar, I don't know. According to you, which polymer can be used as a based polymer, like E. coli used based bacteria to do a material, you mean? So the next question is, how can we use these materials for environmental sustainability? Good question. So actually, it depends. It depends how you how you are using the material. So this is really good. This is a really good question, and this is something that I told you to my students, is that for instance, biopolymers, they are biodegradable, and they are also from renewable resources. For sustainability, environmental sustainability, what for me it's important, there are two things, and these are two concerns that I'm really thinking about. And once I had a sociologist working with me, because in my research, I explore and exploit marine biopolymers. And the thing is that, okay, but we use these biopolymers that are biodegradable, so they can be degradable by nature easily, and to be transformed again in nutrients, to regenerate again other living organisms, and again new biopolymers. The problem is that we need a lot of bioplastic or bio-based plastic or materials. And also regarding the properties, we can't replace all synthetic polymers. for sure, but there are someone that we can. But the thing is, for me, there are 2 points that are very important. One is how, what is the point until when we can exploit these bioresources? Because if we exploit it like we did with fossil resources, so now we are missing it. And so the problem is that we will have a big impact on the biodiversity. So this is very important. Okay, from my side, I'm trying to use waste from fishery industry. But the problem, and sometimes one of my concerns is, okay, but if we develop really a material that it's perfect, and then all the industrial, they want to produce it, and then we will exploit a lot of the biodiversity, you see? So this is a problem for the sustainability, eco-friendly sustainability, if you think about the biodiversity. And then there are another point, is that these biopolymers, as we extract them from nature, we can't use it to do materials like so there are some ones that we can use like this to do materials. But the thing is that we are also modifying it in the lab. So the question is, if we do chemical modifications, are we changing the properties of these materials, but are they biodegradable after? How long they will, but how long we need to the completely biodegradation? And the other point is, during the biodegradation, if we are also doing some toxic molecules that are diffusing in the environment. So these are points that I think we need to think about this environmental sustainability. So biopolymers, yes, for sure, but we have a lot of questions to think about. And it's our responsibility, because we are researchers. So we need to think also, we are researchers, but we are citizens also. So we are part of the society. So we need to think. And I think that we need to work with biologists, with sociologists, so with other colleagues to have to do this well, sustainability.
Sorry to interrupt you, ma'am. Shall we move on to the next section?
Okay.
Because the students are more enthusiastic to discuss with you. The students are never ending. We have Professor Abu Thomas.
Yeah, waiting.
Yeah, waiting here. So shall we go to the next session, ma'am?
No, if there are some more questions, let students ask. What do you think, Hari? We have no, because a lot of your lecture generated a lot of interest, Susanna.
A lot of interest,
Yeah. I mean, our time is limited, right, Hari?
Yes, sir.
Okay,
It's already 45 minutes of discussion now.
Okay, perfect. Yeah.
So thank you, Professor Susanna, for your lecture on starch. It was very eye-opener. You have touched all the details. Thank you so much for your patience also for answering all the questions. I think the students are all happy to interact with you.
Thank you too.
Thank you, ma'am. Ma'am, I have a request. Can you share the presentation or something for the students so that?
Yeah, I will send you. Yeah,
okay.
Yeah,
Okay.
yesterday and today. Yeah, for sure.
Sure, ma'am. Thank you so much, ma'am.
Thank you.
Thank you.
Good. Thank you.
So next we have a session by Professor Abu Thomas. The topic of session is circular economy, new opportunities in sustainable nanomaterials and polymer bio-nanocomposites. But before that, I would like to, I know there is no need of any introduction for Professor Abu Thomas, but I would like to introduce a few words for Professor Abu Thomas. He is an outstanding leader with sustained international acclaims for his work in polymer science and engineering, polymer nanocomposites, elastomers, polymer blends, IPN, polymer membranes, green composites, and nanocomposites. Dr. Thomas' groundbreaking inventions in polymer nanocomposites, polymer blends, green bio-nanotechnology, and nanomedical sciences have transformative differences in the development of new materials for automotive, space, housing, and biomedical fields.
Thank you. Thank you, Hari, for coordinating this exciting GIAN program. And thank you, Susanna, for taking the students to a world of bio-nanomaterials. And I'm glad to note that there were many, many questions to your talk. That means you are a very, very interesting speaker. Thank you so much for taking time in your busy schedule. We all like you to be in Kerala. Unfortunately, you're so busy.
Yeah.
Maybe next time you should make it. We have the funding, you know. Next time,
I know.
visit us.
The first semester, it's really
Tightly
complicated. I
packed,
have
know,
almost my courses on the first semester,
I
but
know,
for me,
next time,
I would, yes, because for me it will be a really, really great pleasure to share with you all these things and to work with you in the lab.
Sure. And I'm glad that, you know, my job is much easier because you have already taken all the students to the field of biomaterials, the biopolymers. And thank you, Susanna.
No, thank you.
Very good, Susanna. And dear students and the colleagues, friends, you know, I'm going to talk about on biomaterials. Susanna has given you quite a very impressive talk about, you know, starch and different types of biomaterials. I'm going to give you an overall picture of, you know, cellulose, chitin, and starchy materials, where I focus more on nanomaterials, nanostarch, nanochitin, and nanocellulose. And you see, if you look at the American Chemical Society statistics, you know, American Chemical Society has come out with a beautiful review on waste, particularly biological waste. Can you all hear me, my dear students?
Yes, Professor. Yes.
Very good. 1.6 billion tons of agro waste. You see, imagine 1.6 billion tons of agro waste is accumulated every year around the globe. Countries like India, Brazil, South Africa, China, a lot of biomaterials because we have China and India, lots of population. Look at our agricultural waste. Look at our forestry products. Look at our fish bio waste, plenty of bio waste. And these bio waste are not really utilized. If you look at India, northern part of India, you know, the farmers burn all their agricultural waste because they have no other way. So they burn it and it causes a lot of pollution. So we researchers have to really make use of these agro waste into advanced functional materials. That is the core of my talk today. If you look at agricultural waste, dear students, we have sugarcane bagasse. You know, India produces lots of sugar, maybe second in the world ranking. And we have a lot of bagasse, the waste that we get from the sugarcane industry. Wheat bran and rice bran, the southern part of India, we are all rice eaters, and northern part of India are wheat eaters. So we have lots of wheat bran and rice bran, corn cob and wheat flour, wheat and rice straw, and jute, coir, pineapple, hemp, banana, bamboo, whatnot, variety of agricultural fibers. Kerala has a lot of coir. Kerala is a land of coconut trees. We have plenty of pineapple now, pineapple leaf, banana. So all these agricultural waste are not really used very effectively. Look at the fish industry. India has a very long coastal area. And we have a huge fish industry. And we are not making use of all the waste of this material into value-added material. Look at starch, potato starch, cassava starch, all the starch material. Starch materials are also waste material. So my talk today is centered around how you can utilize all this bio waste into value-added material. That is the thrust of my talk today, dear students. I want to again show you a very important table. You can see the commercially important plant-based resources. And you can see wood is number one. And followed by bamboo. You can see the world production and the origin. Cotton lynch, jute, enough, flax, sisal, rosella, hemp, wire, you see. And wood is number one, wood base is number one. So we produce quite a lot of plant-based resources. Now, plant-based resources have lots of advantages. They're environmentally benign materials, environmentally friendly materials. They're cost-effective, they're abundant, they're non-abrasive to machineries, they're fantastic acoustic and thermal insulation properties, high specific properties. It's a sustainable raw resource. And dear friends, these are underutilized. Many parts of the world, these materials are underutilized. And this is really a societal need. The use of plant fiber is actually a societal need. Those who want to know about this, you can see our review on carbohydrate polymers. And look at the classification of natural fibers. If you extract from the stem, we call this bast fibers, flax, enough, hemp, and jute. If you extract the fiber from the leaf, we call leaf fibers. Good example, pineapple leaf. If we extract from the seed or fruit, we call it fruit fibers, seed fibers. Good example is cotton and fire. Grass fibers, good example is palm fiber, elephant fiber. So we have a variety of classified waste. And I have some beautiful pictures of banana fiber, which we have plenty in India. Coconut fiber, plenty in South India, Tamil Nadu and Kerala. Pineapple leaf, oil palm, whose industry is coming up in Kerala, oil palm. Bamboo fiber got a lot of eastern part of northeast of India, and jute, particularly in Bihar and West Bengal, we have a lot of jute. And this is the coir and how coir could be extracted from the husk of it. Now, dear students, I'm going to show you what could be the price of this material, relative price. Relative price, I would put cotton unit price of 100. And you can see the relative price of this material, cotton, hemp, abaca, jute, sisal, coir. You can see the relative price of these materials sort of very nicely compared. You look at straw, the cost of straw, cost of wood, cost of fire, compared to cotton. So we have really very nice post-performance ratio for this fiber. The performance is very good and price is very low. I have a slide here that you can understand the physical as well as mechanical properties of fibers. You can see some of the fibers are very strong. Look at NF. The Young's modulus is 50 gigapascals for NF. Look at the hemp. The Young's modulus is 70 gigapascal. All of them are pretty strong. And look at the tensile strength. They're pretty good. And if you look at density, they're much lighter. Their density is in the range of 1 to 1.5. And if you look at the specific strength, specific modulus, they can beat class. That's the beauty of this. Because the density is much lower than class. Now, I want to show all my students what about the chemistry of this. Look at Basically, this is a linear B-anhydroglucose joined by 1,4 glycosidic bonds. Linear condensation flow. Okay, and you see you have beautiful hydroxide. It's a wonderful molecule for a chemist. You know why? Because you can do a lot of chemical results. You can make it hydrophobic, you can make it hydrophilic. So lots of engineering could be done by using plastic chemical reactions. That's a very important point I want to mention. It's a fascinating molecule for a chemist. And if you trace the history of cellulose, you can see plastic work. Dark studies have been reported. The second component of plant fiber is lignin. It's a complex hydrocarbon polymer with aliphatic aromatic constituents. Okay? And then we have the hemicellulose. This is structure of hemicellulose. So plant fibers, the main components are cellulose, lignin, and hemicellulose. Most of fibers, the highest amount of highest quantity is actually cellulose. But if you look at lignin, if you look at coir fiber has a very high quantity. Lignin has a lot of resistance. Now, I want to show you another important slide, the hierarchical structure of plant. Plant fiber, you see, this is a plant cell. And if you can extract the microfibers, so these are microfibers. Inside the microfiber, dear students, you see a large number of microfibers. You can extract our microfibers. Look at these microfibers have a large number. So you can work in different landscape, micro landscape. The possibility to work in different landscape. Now, this talk today, my dear students, I'm going to talk about today how nanocellulose could be extracted from these microfibers. Nanocellulose is not loss of form. Then you might ask, why nanocellulose is so important? There are a variety of reasons. I can list. One is high water. Nanocellulose is a high water. They're extremely crystalline, good mechanical properties, lightweight. Therefore, the specific strength, specific modulus are quite high. High aspect ratio, 10 to 9 ratio is quite high. Have a beautiful reactive surface. You can do a lot of chemistry on the surface. And you can mix it with the many polymers. Okay. In summary, I want to tell you all my students that nanocellulose is a very important strategic platform for developing advanced, sustainable. Very important. This is a very important nano, which can be utilized, which can be extracted from bioresources. This is extremely renewable. Now, how do you make that? How do you make nanocellulose? You can make use of acid hydrolysis. You can make use of steam explosion process. You can make use of high density personification, cryo crushing process, grinding process, high pressure the extraction of variety of technique utilized for high density personification process, crushing, grinding. So if you look at high pressure homogenization process, what is being done is this process really passing the cellulose slurry. You make the cellulose slurry and pass the cellulose slurry at high pressure into a vessel to a very small velocity. And high velocity and pressure, because high velocity and pressure, the nano fibers are sensible. There are commercial instruments available now. homogenization process. The second process is super grinding. Thanks to Abe and Iwamoto, who's really interested to read the paper, biomathematics. They actually commercialized this. They used a grinding where stacking and rotating stone being utilized. So the cellulose pulp will be transformed into anodes. The mechanism of fibrillation in grinder is to break the hydrogen bond, the cell wall structure. Therefore, the nanoscopic material could be made. I request all the students to read the paper of Professor Abe Iwamoto in biomathematics. You can see the nanoscale fibers made by Abe and he. The next way of making nanocellulose is crushing. You cool the nanocellulose under liquid nitrogen gas. Allow the nanocellulose to swell in water, then you cool in liquid nitrogen. Then you crush it using water and pressure. Because of the application of high impact force, the material is transformed. You must read the work of Alimdar and Sain, bioresource technology, every topic. Then you can see the transmitted electron microscopy feature of cryo-crushed cellulose. The next way is high intensity ultrasonication process, we call this HIUS. In HIUS, this is actually a mechanical process in which an oscillating power is used to isolate. We make use of hydrodynamic forces. So when you use this process, cavitation leads to powerful mechanical oscillating power, and high intensity waves are produced, which transform into nanowaves. Then I want to tell you about the steam explosion process. You can see this is a steam explosion process. The steam explosion process, you put everything into a steam chamber. Media. It's a temperature of 1250 degrees Celsius. Once there is a mass. You continue these materials submitted to this high pressure ionization, application of pressure. Then after a while, you release the pressure suddenly. When you release the pressure suddenly, you create an explosion. Because of the explosion, the fiber is opened up. And the fiber is opened up, and finally, the fiber is cellulose and the hemicellulose and liquid undergo chemical hydrolysis, and you get nanofibers. Let me now tell you what we do in our laboratory. My PhD students collect. The waste from during the harvest time. We go to the agricultural land and collect all. We clean them and chop them into smaller lengths. We do initial alkali treatments. Then we get a slurry. Then we go for acid hydrolysis, coupled to the steam explosion process, 3 hours. Then we go for a homogenization process, around 7,000 RPM for 6 hours. Then we get a suspension of nanocellulose in water. If you take a drop of this and evaporate the solvents under the grid of electron microscope, you see what you get. A beautifully entangled network of nanocellulose. Dear students, we are able to get, when you start from plant fibers, when you do steam explosion and acid hydrolysis, we are possible to get two products. One is the long nanocellulose. It could be 1,000 nanometer length. The diameter could be the range of 20 to 70 nanometer. This is the long nanocellulose where you have crystalline amorphous phases coexist. Crystalline amorphous, crystalline, amorphous, amorphous and crystalline. On the other hand, if you use a very strong acid, all the amorphous phases will be eaten by minerals, and you get short nanocrystals. So we are able to make either short nanocrystals or fibers. The totality of the, synthesis protocol is quite simple. We collect the base, we go for acid hydrolysis, dialysis, or mineralization. Look at the tensile strength of the material. The tensile strength of the material is 10 gigapascal, and the modulus is 130 gigapascal. And these materials have lots of advantages: low force, regular shape, axis ratio, monocrystalline, and now I wanted to show you some interesting feature. You can see the work of Professor Muhammad and group. Professor Muhammad utilized acid hydrolysis. 65-8% of sulfuric acid used for extracting nanocrystal from you can see he could get the short nanocrystals moved by acid hydrolysis. Let me show you what we have in our laboratory. Our laboratory center, we are going to show you the raw fiber. We are subjecting the raw fiber to steam explosion units. Then you go for a bleaching process. Then you go for acid hydrolysis. You repeat the process till you get the nanoscale. Then you go for a centrifugation process. Finally, we get. I also want to show you the enrichment process. You see, if you take raw fiber, the cellulose content is 64. Lignin is around 18%. Then, hemicellulose is 18%. Lignin is 4%. But after steam explosion process, you see, the amount of cellulose has gone up. After bleaching, after acid hydrolysis, you get almost 95. You can increase to 99%. You repeatedly do lots of bleaching process. You can reduce non-cellulose content and you can get a rich content of cellulose. I want to show you some nice pictures of nanocellulose made by my peers. The nanocellulose made from banana stem. These are all nanocrystals. You can see nano fibers made by my coworker from wood flower. You can see the nano cellulose fiber made from pineapple leaf fiber. In explosion process, FESEM picture of nanocellulose from pineapple leaf fiber. Now, see, dear students, we can do a lot of chemistry of the surface. Why do we do chemistry? Because nanocellulose has a polar surface. Therefore, it is difficult to be dispersed in non-polar poles, polystyrene, etc. So, therefore, if you don't do any chemistry, the application is largely limited to aqueous or polar systems. So, to overcome these problem, what we can do is we can do a lot of chemistry on the surface of nanocellulose. So this is nanocellulose. You can go for a cellulation reaction. Now, my coworker is doing cellulation reaction for the car company. You can do catalyzation reaction. You can reduce fluorescent label to do beautiful fluorescent spectroscopy work. You can do TEMBO mediated oxidation process, so OH will be converted into COH or CONA. You can do esterification. You can do polymer grafting. Partial groups could be, polymers could be made on the surface of nanocellulose. There are lots of possibilities exist. These possibilities, these chemical reactions can transform the intrinsic properties of nanocellulose. Now, dear students, I'm going to talk about some of the applications of nanocellulose. It could be utilized for wound healing. It could be utilized for water purification. It could be utilized for sensors. It could be utilized for EMI issues. So I will also give you examples of each application. You know, cellulose nanomaterials in membranes for water purification. The Duke SRP researchers described the potential benefits of advancing the use of cellulose nanomaterials for water purification. We can all look at the Duke University work. So this is, I'm going to show you what we have done. My research group, what has been done. If you look at the commercial water filtration cartridge, they are not very efficient. Most of the cartridges are made of bilateral. The bottom layer is mostly polyacrylonitrile, over which you have a PVD, polyethylene fluoride. We found that commercial filters are not very efficient. In order to increase the efficiency of the commercial filter, we decided to give a coating of nanocellulose. So we have given a coating of nanocellulose at the surface of. Yes, students, once you give the coating, the advantage is the hole size becomes very small because you have a nanocellulose. The system becomes more hydrophilic, so water can diffuse very easily. You can see one of the projects we have done is crystal violet is a very, very important dye in textile industry. But after dyeing, you see many rivers and water bodies are polluted by crystal violet. So what we did is, in order to attract crystal violet by electrostatic interaction, we did some chemistry on the surface of nanocellulose. You can see this is nanocellulose. We reacted with the Meldrum's acid. Once you react to the Meldrum's acid, you can introduce CO minus group on the surface of nanocellulose. The idea is to make nanocellulose more of CO minus group, make nanocellulose electronegative. Okay, that is the purpose of putting the CO minus group on the surface of nanocellulose. This is a clean chemistry. Green chemistry, clean chemistry. So we introduced the CO minus group on the surface of nanocellulose. Now I'm going to show you the results we got. Absorption capacity of the crystal violet dye, milligram per gram, as a function of time. We went up to 2 minutes. Look at this is the commercial filter that you get in the market. You see, behavior of commercial filter goes up and sort of leveling off. This is the commercial filter coated with unmodified nanocellulose. You can see the filtration efficiency has gone up. It was 1 or 1.5 became almost 5 with the coating of nanocellulose. Yes, students, look at the blue one. This is PVDF modified with chemically modified nanocellulose with the CO minus. Once you put CO minus group on the surface of the filter, CO minus group on the nanocellulose, the absorption capacity has been remarkably increased. Now the value is 4. So this is a remarkable increase in the capacity. Interesting. With the impure water, this is the purified water. So then you might ask how this happens. Look at this is the modified membrane. The top is actually nanocellulose coating. And when you purify water through this membrane, you see all the crystal violet is stopped on the top surface of the filter. You might ask why crystal violet has been stopped on the top of the filter? Because of the electrostatic interaction. This is the chemically modified nanocellulose, and this is the crystal violet. And there's electrostatic interaction between crystal violet and nanocellulose. Oxygen of nanocellulose, modified nanocellulose interact with the we can explain the affinity of crystal violet with nanocellulose. Let me show you another interesting work. You see, this is a work of Professor Ma. Professor Ma looked at the commercial filter to remove ultraviolet dye. This is the performance of commercial filter. Absorption capacity milligram per gram as a function of time. Look at the commercial. But when you utilize nanocellulose, you see nanocellulose, nanoblister, you see there's a remarkable enhancement in the absorption. You can see this is the pure water. This is pure water purification. Dear students, my coworker Deepu and I were really interested to look at the suspended particles here. Probably you all know that water today has a lot of suspended. It could be oxides, it could be metals, so we decided to just filter water and remove all the suspended particles. You can see these are the suspended particles. So we coated the commercial filter with the nanocellulose. We don't do any chemistry, we coated with the nanocellulose. Then they filtered the water with a lot of particles. For example, this is a model system. Water actually filled with iron oxide particles. So this is a model system, impure water. And if you look at the transmittance of this is almost zero because of the. But then we filtered this water to the filter. The top surface is PVDF coated with. You can see all these iron oxide particles have been stopped at the top surface of the filter. How? By absorption. This is the bottom part of the filter, which is made of PVDF. So we have shown that we can remove all these sort of something very interesting. And you can see this is the pure water and this is pure water. Now, dear students, I'm going to show you some work of the Finland group. The Finland group, they utilize nanocellulose for removing oil from water. Probably all my students must be reading in the newspaper that seawater, river water are increasingly contaminated by oil, particularly the leakage from the ship. Oil tank, there could be leakage. Sometimes you must be reading the sinking of ship in the sea. So therefore, seawater is increasingly contaminated oil. Therefore, it affects the ecosystem. So what the Finland group did is, that is something very interesting. That dear colleagues, dear students, please look at. The Finland group, they made nanocellulose. How do you make it? You have 1 or 2% of nanocellulose in water. For a freeze-drying process, you can make beautiful. Aerogel is highly porous. And then what they did is they did something interesting. They made a coating of nanocellulose with hydrogen dioxide. You see, they made a coating. These dry nanocellulose aerogels were coated with. Why did they do the coating? Because to make it hydrophobic. Once you give the coating, you see these fibers become hydrophobic. And these fibers are extremely porous. You can take the coating. So once you make the surface of the aerogel hydrophobic, you put in seawater. And when you put in seawater, what happens? This red is the oil. All oil is absorbed by the aerogel because it's hydrophobic. And it's porous for the capillary reaction. All the oils could be absorbed. As a result of these aerogel undergo swelling. Once it is swollen well, once it absorbs all the oil, so there are different ways you can squeeze it out. You press it and take out the oil and recycle. One way. Second way is don't for pressing. You can burn the oil. Two ways you can do. But I always recommend for recycling process. Now, I want to show you the use of nanocellulose for heavy metals. Probably you all know that water is increasingly contaminated with heavy metals, heavy metals of zinc, lead, mercury. It's a big problem worldwide. But we have more problem in the northeast of India, heavy metals. We have a big problem of heavy metals. So you look at this slide, this is equilibrium absorption of milligram per gram of chromium as a function of concentration. You can see a beautiful absorption of chromium. This is chromium 6. I wanted to tell you one thing. This particular chromium 6 is electronegative. So to accelerate the absorption process, you can react the nanocellulose with polyvinylamine. You react with polyvinylamine, you can introduce massive amine group. Massive amine group. You can introduce massive amine group on the surface of nanocellulose. You can introduce massive amine group on the surface of massive amine group. So these amine groups are positive, and chromium 6 is negative. So you can have such interaction, and this is the way the chromium is removed. Similarly, you can remove nickel, you can remove cadmium, you can remove lead. So all these impurities could be removed. Nickel could be removed, cadmium could be removed, lead could be removed. And some of them could be removed without any electrostatic interaction. Some of them could be removed by electrostatic interaction. But probably you can look at this slide. Our great grandfathers, they used to use rice straw fiber. See, look at this. I remember my great grandfather used to purify water using rice straw fiber because we have a lot of rice straw in our home. So he used to make a bed of rice and then purify water. You see, 50% absorption. If you go to micro length scale, you get around 70%. If you go to nano length scale, you look at 90%, 95%, sometimes very close to 100%. So nano is offering lots of things. So this is something very interesting. Nanocellulose could be removed, nickel, cadmium, and lead. And these nickel, cadmium, lead are electropositive. And nanocellulose have intrinsic negative charge. Nanocellulose have intrinsic negative charge, so that you can have electrostatic interaction between cellulose and this. Now, dear students, we can remove bacteria and viruses. Bacteria and viruses could be. Bacteria can be found in large numbers in we all know that these microbes causes cholera, dysentery, typhoid. Okay? So you can remove bacteria and viruses. Bacteria are responsible for abdominal pain, gastroenteritis, virus, cold, diarrhea, respiratory infections, hepatitis. So a lot of complications. So what we can do is we want to remove bacteria. They are in the range of 1 micrometer size. So you can separate them by single size, so they are bigger in size. So you coat the commercial filter with nanocellulose and filter it. All this bacteria can be stopped in the process of the But dear students, what do you do viruses? So viruses are difficult to be separated. What we do is we do some chemistry. We react to the polyvinylamine. When you react to the polyvinylamine, you can introduce massive amine group on the surface. And then you can have electrostatic interaction between the positive charge amine group and the virus. So this way you can remove viruses. And you see the logarithmic reduction value for bacteria 6. logarithmic reduction value for a virus could be greater than 4. So you can have a very good removal of viruses. And you see the pore size of the membrane, something like 0.78. Okay, that's the pore size of the membrane. Quite small so that you can remove yeah. Now, the biomedical applications. Biomedical applications also you can utilize. You can see a gentleman suffering from high degree of burn from his face. And you can see his face is completely covered with nanocellulose gel. Nanocellulose gel, intrinsically they are high porosity, fast tissue regeneration. Initially, you can also introduce drug to this platform, and drug could be slowly. Now I'm going to show you some nice work of university and Sao Paulo University in Brazil. My Brazilian counterpart was very active in synthesizing urethane is a very important material for manufacturing. So he, in cooperation with my Brazilian counterpart, we made a polyurethane cellulose nanochemistry. You can see the lead polyurethane strength is 17.5. E modulus is 37.5. Dear students, you add 5% of nanocellulose made from pineapple fiber, 17.5 megapascal becomes 37.5, E modulus becomes 990. It's a quantum jump. 17.5 37.5 becomes 990. It's a quantum jump in the quantum jump in this language. Eventually, dear friends, eventually he made nanocellulose polyurethane possible. And all credit goes to my Brazilian collaborator. He worked with the Brazilian hospital and he made all this aqua. Okay? And he also recommended that vascular processes will be made of very good. Your friends, nanocellulose is an excellent platform for you can see these are all the food. These are clothes, the new platform where you can introduce antibiotics. Antibiotics to be slowly released to the see a beautiful work of Sebastian Morris. Sebastian Morris made gel. This is bacterial nanocellulose. Then he introduced the antibiotic into this. And Professor Sebastian, he looked at the relative release of the antibiotic. You can see a beautiful release of. You can see this could be easily. It's an excellent platform. Everybody applications. Let me now show you the work of Professor Liu. Professor Liu and Liu, what they did, they made aerogel of cellulose and collagen. Then they did some slinging process using dialysis. Look at these nanocellulose collagen. Aerogel was extremely porous. Then he made nice interesting material. And Professor Liu reported that the water holding capacity of these aerogel is something like 4,000. High degree of water holding capacity. And the density of this material is 0.02 to 0.03 gram per centimeter. They're extremely light because highly porous. And look at the porosity they have calculated. The porosity is 5 degrees. And Professor Liu and colleagues tested that these materials are very, very nice because you can absorb all the pus and the water. It becomes very dry and only is extremely fast. It is an excellent fantasy for innovation. I want to tell you that nanocellulose is an excellent reinforcement because you give nanocellulose coating electroscopics. You can see this is the pan membrane. The pan is interesting for mechanics, but the mechanical properties are very small. You give a coating, one-side coating, see on pan, you see the spine goes up. You give one more coating, you see high increase in the mechanical. This is basically people say this argument. I should tell you something. During the COVID, we have designed a structure. It's in this pasta. Outer layer is made of yes, the layer is innermost layer is made of so the outer layer has another important property. The outer layer is extremely hybrid. So given a polyelectrolyte coating, that outer layer has very high level of. Therefore, these layers can repel coronavirus. And you see, you wear the mask, the outer layer can repel coronavirus. If at all some coronavirus getting in, the layer is engineered such a fashion. It has positively charged. These nanocellulose can make electrostatic interaction. All these viruses. There is very comfortable. You see, this is a design. Gesture with anti. But also coronavirus. Also made by. Now, I want to tell you some important application of nanocellulose as sensing. Nanocellulose is a very good platform for sensing application. See, what has been done is this is nanocellulose, and this is CNG. You combine nanocellulose and CNG together, you make a construction. Okay, you combine both, and then you mix with the natural rubber latex. Kerala has plenty of natural rubber. So you mix with the natural rubber latex, so you get a beautiful dispersion. You go for a mixing process, coagulation process, and hot pressing. Once you do the hot pressing, you get a beautiful three-dimensional structure of cellulose and CNG in natural elements. Okay, this is a 1:1 hybrid construction. Now I want to show you something interesting. If you look at this slide, you see this is CND, CNC, and natural rubber. CND, CNC, and natural rubber. very nice distribution. We put CND alone in natural gas, a lot of accumulation. So my point is, we combine CND and CNC, carbon nanotube and nanocellulose together. If you put in natural gas, you can get very high degree of dispersion. And I can show you this interesting slide. This is the sensing action and the conductivity. If you put the CND in natural rubber, you get a percolation process beyond CND natural. Normally, CND is very difficult to be disposed of. And dear colleagues, if you combine CND and CNC together, hybrid configuration, look at this. At 1.5 volume fraction of volume percent of CNG, you have a very nice. So a 50-50 combination can shift the percolation to lower concentration of CND. Something very interesting. The CND dispersion is very good, and you can have fantastic networks. You see, the mechanical properties are very good for the hybrid structure. So this is the best one. You see, look at the sustained behavior. The hybrid configuration of CNC and CND national. And now I want to show you the strain sensing. Look at this. You stretch it, and this black is actually CND, CNC in natural rubber. A 1:1 combination. You see, there's a large increase in strain level. You can see there's a large change in electrical conductivity. But you look at the other combination, they are not very efficient. This particular combination, 50:50, CND and CNC, is a beautiful material for strain sensing application. Okay. And now you see the strain sensing. You can see you stretch, you release, you stretch, you release, you stretch, you release. You can see the variation of electrical conductivity. Decrease, increase, decrease, increase. So this could be a very good sensor. Similarly, dear students, we can combine nanocellulose and reduced graphene oxide. We can combine both. This is CNC and this is graphene oxide. You mix them together, mix with the natural rubber latex, and then you coagulate and dry it. You see what you get. This is RGO. This is RGO CNC. 0 hour. After 24 hours, if you keep it, you see all the RGO is settled. But look at RGO CNC is extremely stable. So my point is, when you mix RGO and nanocellulose together, the dispersion becomes more efficient because the RGO interactions could be suffered. They don't undergo any agglomeration. And now I want to show you the percolation process. If you use RGO alone, reduced graphene oxide alone, you see the percolation process beyond 0.5, 0.75. But if you combine both RGO and carbon cellulose nanofiber, look at this. The percolation is shifted to lower concentration of RGO. This is something interesting. And then another important data I want to tell you, these hybrid system, this is pure natural. These hybrid system show very high mechanics. Hybridizing CNC and nanocellulose together and putting into a polymer matrix has a lot of relevance. If you mix two fibers together, two nanomaterials together, you get better dispersion in the polymer matrix. And you can see the sensing action. You can see the relative resistance. Relative resistance goes up. Relative resistance goes up means conductivity is decreased. Because when you stretch it, the conductivity is decreased. And you touch it, the conductivity is, you can see it goes back. And you can see the number of cycles, first cycle, second cycle, third cycle. This is pretty good. You can see the RGO, this RGO NR dispersion was not good. But if you look at RGO, NR, and nanocellulose, get very good dispersion. Dispersion, really very good. And you can see the responsivity time relationship of nanocellulose, RGO action. You see, look at this. This is relative resistance as a function of time in toluene, in chloroform, DMF, and you see, toluene is a very good solvent for natural rubber. So the toluene is easily absorbed by the natural rubber, and the network is destroyed. So we can see the relative resistance increases. Similarly, chloroform is also easily absorbed by natural rubber. So the relative resistance increases. But look at the DMF and acetone, they're not very good solvents. Dear friends and colleagues, nanocellulose is a very good platform for energy storage applications. We can make paper-based electrodes. We can make aerogels for supercapacitors. We can make anodes for lithium-ion batteries. We can make beautiful aerogels. So a lot of nice applications. This is actually the work of Professor Wang and Yu. Professor Wang made aerogel. Just look at this aerogel made by. Then he went for pyrolysis of the aerogel. He pyrolyzed it, and he get a carbon aerogel. This carbon aerogel is an excellent anode material for lithium-ion batteries. An excellent material for because this is extremely porous. Therefore, the electrolyte reaction is very strong. So, Dipping Wang suggested that this is a very good material for lithium anode for lithium-ion batteries, and extremely porous. Therefore, the interaction with the electrolyte is very active. Now, I want to show you some recent work on the production of nanocellulose-based aerogels. You can make beautiful aerogels out of nanocellulose. I wanted to cite the work of Mr. Gopakumar Pai. Gopakumar made nanocellulose from the stem, then he introduced aniline, then he went for in-situ polymerization of aniline. And he made aerogel of nanocellulose. This is the, when you add aniline and CNF, this is the morphology, and this is the aerogel. Now, this is the SEM of the nanocellulose paper, and this is the SEM of the pani CNF fiber. And you can see that this is extremely conductive. This is extremely conductive material. Very much conductive material. And this is also lightweight. You can make lightweight electromagnetic shielding material using nanocellulose and pani material. And the manufacturing is quite simple process. And this could be a very nice material for EMI shielding applications. EMI shielding applications, you see, EMI shielding in the range of 20 or 22 or 25 or 24 dB could be achieved by using nanocellulose and pani. You polymerize the pani in presence of nanocellulose, and you made beautiful EMI shielders. And if you look at this EMI shielding application, basically the shielding is by absorption process. So most of the unwanted energy is absorbed by the shielding material. And the reflection is only 13%. Major component is absorption process, around 87% absorption process. Dear students and colleagues, I have only a few more slides to finish. My research group is active in chitin, particularly nanochitin. Nanochitin can be extracted from the craft shell or corn shell, and they're quite nice ways of making nanochitin. So what you do is, this is the structure of chitin. Chitin is very similar to cellulose, but you have a fascinating NSCOCH2 group that can do a lot of functionalities. And this is second most important biopolymer, the crust of the earth. So what I want to tell you is, making craft shell from craft shell or corn shell, if you do some simple processing, you can make either nanocrystals of chitin or nanochitin fibers. If you use the less harsh extraction process, you get nanofibers, particularly if you use light acid. If you use strong acid, you get nanocrystals of chitin. So our group is very active in extracting both nanofibers and nanocrystals. So I wanted to show you just one or two reactions. One reaction. See, look at this. This is a nanochitin, the structure of nanochitin. Nanochitin is actually hydrophilic material. But yes, students, we can make it hydrophobic. You react with bromhexadecane. React with bromhexadecane. Bromhexadecane can interact with the nanochitin, and then you can introduce beautiful alkyl group. With this simple chemical reaction, you can introduce and you can control the number of alkyl group on the surface of. If you put a lot of alkyl group, system becomes extremely hydrophobic. So you can engineer the hydrophilicity, hydrophobicity. And by controlling the number of alkyl group, you can make nanochitin of varying contact angle, varying degree of hydrophobicity. This is a neat nanochitin whose contact angle is 86.5. As you put more and more hydrophobic group by this chemical reaction, you can go up to super hydrophobic surfaces. This contact angle is 158.2. And you can follow the reaction between a bromhexadecane and nanochitin by NMR spectroscopy. And you can see these TIA images of the nanochitin and modified whisker with the bromohexadecane. And you might ask, what could be the utility of this material? This is an excellent additive for oil. Okay? This material is an excellent additive for oil because to control the viscosity of the oil. So this is a beautiful additive for modifying the properties of hydrophobic oil. So by doing a small reaction, we found that the material becomes very, very important for controlling the viscosity. But some time ago, my PhD went to Slovenia, and we did some work with the Maribor University. What we have done is, my Maribor collaborator was using indiocarmine dye for water purification. Sorry, using PVDF to remove indiocarmine. Indiocarmine is a pollutant material in water. So she was using PVDF to remove indiocarmine. But we look at the efficient separation. Then I suggested, why can't we combine PVDF and nanochitin? Then we made a hybrid configuration by electrospinning. We introduced nanochitin with PVDF, and we went for electrospinning process, and we made membrane, and then we tried to purify the water. And we found that indigocarmine removal was increased to 88.9% when we used a configuration of PVDF and nanochitin. So see, a lot of possibilities of combining nanochitin with other polymers. Then you can increase the separation efficiency. This is something very interesting. And dear colleagues, students, the last part of my talk is more on starch. Susanna has talked to you about starch, structure of relationship, applications. She is continuing the lecture. Starch is also a fascinating material. But yes, students, we don't touch the food sector. Starch is very important. If you don't, what we do is we collect the waste starch from agricultural lands. No food is touched because the huge population out there, very close to 1 billion, so we cannot use any starch. Very important. So what we do is we collect the waste material in agricultural lands, like potato, like rice, wheat, sorghum, tapioca. We have plenty in Kerala. So during the harvest time, my PhD students go to the harvest ground, field, and collect all the waste from it cannot be used by human beings and animals. We collect it, and then what we do is we do an washing with acid. So by washing with acid, we remove all the phosphorus part. And dear students, we can have very nice nanocrystalline powder of starch. So you can easily make nanocrystalline starch powder. You can see some electron microscopic picture of nanocrystalline starch. And when you do the preparation of the nano starch, you can control the concentration of acid, duration, nature of acid. So by doing this, you can control the properties of material. So what we have done, my dear students, we made nanocrystals of starch from waste potato, and then we characterized them. Then we put them into natural rubber latex. Kerala has plenty of natural. So this is neat natural. Look at this. By putting 5, 10, 15, and 20% of nano starch, you see, natural rubber is transformed into a high modulus. High modulus. Rubber is very, very flexible. You can see the flexibility is reduced and becomes very strong material. And the strength and modulus of natural rubber has been increased quite dramatically. Now, I want to make a few concluding remarks. My message is don't throw away the agricultural waste. They're all value-added material. We can transform them into advanced functional materials. Nanocellulose you can have in nanocrystals or nanofibers. You can control the morphology very well. And you can utilize them for water purification, utilize them for wood healing, utilize them as a nano reinforcement for polymers. We can utilize them for drug release. You can utilize them for energy storage, EMI shielding, sensing, variety of applications. Similarly, chitin is also a very fascinating material, second abundant biopolymer, the crust of the earth. By an acid washing, you can make nanochitin fibers or nano whiskers, and a lot of applications, reinforcing filler. You can use it in water purification, tissue engineering, heavy metal removal, environmental remediation process. Finally, the starch, we don't touch the food sector, we collect the waste starch. You can make beautiful nanocrystals. You use a reinforcing filler, you can use it for packaging, variety of applications for the nanocrystals of starch. So I'm posing a question to all my students who are attending this seminar. Can we become a zero-waste planet? The answer is yes. We all have to work hard and transform all our agricultural waste into value-added. And we have to preserve our ecosystem for the coming generation. So this is a very important message to all my students, colleagues and friends. Don't throw away the waste. Transform them into advanced functioning. So I would like to thank all my co-workers who worked the night. Government funding, Professor Rao is acknowledged for the nanomission funding. I would like to thank ISRO, CSIR, DBT, AICT, EBC, DRDO, DIG, DRNS, international funding, Fix France, India, Sweden, India, Slovenia, TWAS funding, DuPont, GIAN COURSE, many cooperation with other countries, surface fleet check, initial funding, part of Apollo, Fiat tires recently, and all my problems. Daniel helped me a lot for making this platform for water purification. My colleague Nanda Kumar, so is quite active in polymers. Our has made a lot of exciting promises for medical applications. Matthew, we did a lot of work on from food power, Hawkins, Maya Johnson from South Africa, Didier Ruxel from France. Iran did a lot of XPS measurements. Selvar also did a lot of stuff. Thank all of them. My group in the campus, my chemistry group, is our group in Nano. So the Nano group, and this is a journal I am editing. This is the editor of I'm the editor of nanostructures and nano objects, site score 11.9. Elsevier journal, no pay charges. So those who work on nano, please submit your articles to this journal. And we have two conferences coming up here. International Conference on Macromolecules, 12, 13, and 14. International Conference on Nanostructured Materials and Nanocomposite, May 10, 11, and 12. Thank you. Most welcome for this conference. And thank you so much. We are also publishing quite a lot in the area of nanomaterials. You can see our publication track record. We are very active in books, editing books. We have many books in pipeline, and you can see our book on nanoscale materials of water purification. So thank you very much for patient hearing, and I welcome you all to my campus. Please visit us in Kerala, some nice pictures of Kerala. We have beautiful canals, opportunities for cooking, sports, waters, recently plantation, Kerala, Kathali, thank you. You have every reason to visit Kerala. Thank you so much. I welcome you all to Kerala. And thank you for participating in this GIAN lecture. Thank you.
Thank you so much, Professor. Thank you so much for your very in-depth lecture. I think we can have few questions because we are already running late. If there are any questions, please type in the chat box or you can unmute.
If there are any questions, Any questions? Any questions in the chat box? Yeah.
Sir, there are some questions in the chat box.
Yeah. Yes, that's from here. Hari is there. Hari, where is it? Hari, which are my questions? Some are to.
I will read out your questions.
Yeah.
All these are very thin and can bear small magnitude of forces. Can you please tell us about nanobiomaterials which can withstand large magnitude of forces and can possibly have engineering applications such as biomaterials, which can be useful for heavy load carrying bridges.
Yeah, you see, nanocellulose and nanochitin, particularly nanocellulose, we are now going to utilize it for tire engineering. I have a project with Apollo tires. So we are going to utilize it for tire tread and inner liners. Definitely you can utilize it. So many polymers, look at our hot wall. We have made good reinforcement on polyurethane. Therefore, nanocellulose is a very good reinforcing material. Because look at the modulus of this material, 130 gigapascal. So no polymer can beat them. Therefore, they are very good reinforcing agent for polymers. What's the next question?
Also, the next question is, sir, can we modify sensor substrates with biopolymers? If you could, you discuss a few.
Definitely, you see, you can make beautiful sensors out of nanocellulose. I have shown that if you make a combination of nanocellulose and the CNT together and put it in natural rubber matrix, you can have a very good dispersion of CNT. CNT forms in it, low concentration of CNT, and then you stretch it. You know, when you stretch the rubber, network is destroyed. So it can act as a very good sensor. Similarly, graphene, combination of graphene and nanocellulose in natural rubber biopolymer, you can make beautiful sensors. So the sensing is good way, particularly we focused on strength sensors. Chemical sensors also we can do. We can use swelling in toluene, THF, acetone, all this. So we found that when you put toluene as a solvent, toluene enters into the rubber matrix and the network is destroyed. So this could be a very good sensor for strain sensing. It could be a very good sensor for sensing solvent. Thank you.
So one more question. In rice straw, while we treated with NaOH, it forms sodium orthosilicate. How do we treat it?
You see, rice straw, what we do is rice straw, you treat with sodium hydroxide, and sodium hydroxide will destroy some of the hydrogen interaction. So what you get is the oils to be more exposed, and you get some ONA. Sodium cellulose you can make. And these are initial treatments. And then you go for the further treatment with acid, then you can remove all the lignin initially. Okay, this is the protocol we do. Thank you. Thank you so much. Thank you for your patient hearing. Thank you, Hari. You can wind up.
Thank you, Professor. Thank you so much. I think there are no more questions. Then we can wind up today's lecture. Thank you, Professor Sabu Thomas, and thank you, Professor Susanna, for your lecture today. If there are more questions, you are always free to ask through emails.
Thank you. Thank you so much. Thank you.
We can meet tomorrow at 1 p.m.
Thank you.
Thank you so much.
Thank you,
Professor Susanna.
Thank you.
Thank you.
Thank you, Susanna. Bye-bye.